All right, students, we're going to pick up exactly where we left off last time with osmoregulation. You should remember this. They're going to show you a picture of the paramecium that is going through this. Right, so there you can see the osmoregulation, getting rid of it and then taking it in. As we talked about before, cell walls are going to help maintain the water balance within a plant cell. As it's hypotonic, it's getting what we call turgid, so it's really going to be nice and firm, whereas if it's isotonic, or in this case hypertonic, it's going to go flaccid or limp. All right, in that hypertonic environment, we're getting something called plasmolysis, which is going to kill the cell. There's not enough water. In fact, it's nothing that is going to help the cell survive. And that's plasmolysis. That happens in regular cells, animal cells as well. All right, so we talked about diffusion, and now we're going to use some proteins to help with that diffusion. That's called facilitated diffusion. So this is just transport proteins that are going to help aid movement of bigger molecules. So that's going to be your aquaporins. It's also going to be some bigger things that have gates on them that are going to have a specific kind of molecule that are going to go through them. But those gated channels and that facilitated diffusion, again, now we're passing through a specific part of the plasma membrane, the phospholipid bilayer, and all that is doing is helping things go on into the or out of the cell. And this again still doesn't require any energy. So here's an example of that. We're getting a solute, and these things are going in, so coming in and then going out. So again, the protein is going to kind of change its structure, allowing things in and out of the cell, as you can see in this picture. And you saw that on the previous picture. It's allowing those things to kind of bind and then come inside. There are some diseases that can cause some issues. As you can see here, again, no energy for this. Facilitated diffusion is still passive. It's moving down the concentration gradient from high to low. But there are some that move them against, but those, as they're moving against a concentration gradient, are going to require some energy. All right, and that's what we call active transport. So if we move something against the gradient, going from low to high, that's going to require energy, of course, usually in the form of ATP, and that active transport is going to be by very specific proteins, and they're going to be embedded in the membrane. These are going to be transmembrane proteins, those integral proteins that we've already discussed. So one of the biggest ones that we talk about is the sodium-potassium pump. This is, of course, very important for brain function because it's sodium-potassium that is going to be driving the nerves to do their function. The brain is going to demand a lot of sodium-potassium. And as you can see, this is how that works. It's going to be 3 sodium ions to every 2 potassium ions. They bind to the intramembrane protein, and then they're going to move. It's going to move the sodium out. We're taking ATP that's going to go down to ADP. Need to move this up the against a concentration gradient, and we move the sodiums out. Then we move the potassiums in. And the cycle continues to repeat itself, as you can see here. Again, passive, no energy, active, energy. All right, and then this is all about voltage differences. So this is a membrane potential, and that's also going to be an ion pump, again requiring a certain amount of ATP because that voltage is going to be created by positive and negative ions, and that difference between those ions is going to kickstart that reaction with those proteins that are going to drive that membrane potential and change it so that we have things moving back and forth as long as we have that voltage difference between inside and outside of the cell. All right, and those combined forces, it's called an electrochemical gradient. That's driving that diffusion. Again, that's only going to drive that diffusion as long as we have a difference between the charge on one side or the other. All right, and then we have these wonderful electrogenic pumps. They are transport proteins. They're generating that voltage across the membrane. Sodium potassium is going to be one of those. That one's going to require a lot of energy. And then, of course, another electrogenic pump for plants, fungi, and a lot of bacteria is a proton pump. So if we're doing protons, it's essentially an ionized hydrogen atom that now, of course, has lost its electron and is now just a proton. So here's that in in function and in practice. So you're seeing the energy right here. We're going to move this again. This is going from low to high, so that's why it requires energy. It's a form of active transport from the cytoplasm out into the extracellular fluid. And then, of course, we can do a little bit of coupling as well, active transport of a solute going, also driving transport of another one. So this is proton, these pumps working together, these proteins working together. This is going to happen a lot in the plants, and it's not just going to drive your protons, but it's also going to transport various nutrients into the cell. Again, hey, why use extra energy when we can activate it in another way? So here's that. So we're getting sucrose plus hydrogen, so we're transporting sugar, and our proton pumps, it's actually activating other things going on. So we're moving the protons out, we're getting the sucrose in, and still requiring energy, but these things are working in tandem with one another. So similar idea between the sodium-potassium pump, but this one is a proton and sucrose that are working together with one another. And then, of course, there are other things that are coming in and out of the cell. Small molecules are going to go back and forth, and that's going to go through those transport proteins that may or may not require energy. Big O polysaccharides, which aren't going to move very easily unless they can go into the membrane, those are going to require energy. And then, of course, if we're moving a lot of stuff in and out, that's going to require lots of energy, right? Bulk transport, the more you move in, the more you move out, the more energy it's going to require. And then, of course, if we're transporting things out of the cell, that has a specific name. It's called exocytosis. We're moving things out of the cell. Very simple. There's a lot of different parts of the cell that are using exocytosis to get rid of things. That's also going to include various forms of garbage that the cell no longer needs. Those are going to be moved out because that'll keep the cell healthy. If we're moving things in, like your good old bulk transport, that's going to be endocytosis. Again, endo meaning in and exo meaning out. And these are going to form vesicles as they come in, so that's actually going to be part of the plasma membrane. And we're moving things in. There are three types. We have phagocytosis, which is what white blood cells do. They eat the cells. We have pinocytosis, which is cell drinking. And then there's ones by various proteins that are on the phospholipid bilayer, and that's going to be receptor mediated. So again, that's going to be moving things in and out, and it has to be done so with a specific kind of transport. So it's looking for specific shaped molecules in that case. So in this case, we got phagocytosis, eating a cell, and usually fusing with a lysosome, and they'll digest the extra particles. This happens a lot with white blood cells. They're really good at doing this, and can actually be kind of interesting to watch, but sorry, no videos other than the one that you're looking at here. So here we have those processes that you can see here. And then your things moving in and out. We have that drinking, and you can see that from the microscope, again, 0.5 micrometers that you're looking at there. And then we have these various ligands that are, again, specifically binding to a specific spot. Let's look at that receptor-mediated endocytosis. Here we go. There's the receptor. We got things binding. That binding, of course, we're moving our phospholipid bilayer, which is then becoming our transport vesicle that is going to go where it needs to, according to the instructions from the cell. All right. And again, this is going to be a chemically mediated process. All right, so here's your different types of active transport, passive transport, and these are the questions you guys are going to answer at the end of the day, and that is it for the video. I hope you have enjoyed your journey into the cell membrane and all the things that it does and mediates to maintain homeostasis within the cell. And we will pick up on a lab on Friday, and I will see you guys then.